So I'm going to go through the review that I would have given you in class with the clickers, but I want you to definitely study all your notes and use the review you create by focusing on every single topic in the class. So responsible for the model of DNA, Watson and Crick. Let me change colors. X-ray displayed, that would be Rosalind Franklin. Transforming factor was protein, is Griffith. Used mice to discover that the transforming factor was DNA, so now it's genetic, was Avery. And then Hershey and Chase piggybacked on that with the bacteria, which is right here. I'm sorry, with the virus that affected the bacteria. So it's Hershey and Chase. This is Chargaff. A equals T, C equals G. So he discovered that all nitrogenous bases in a DNA model, all the I adenines will match up with the thymines, and the cytosines will match up with the guanines, and their compositions will be equal. That's what the equal sign's for. So nucleotides are made up of three things: a phosphate, a 5-carbon sugar, which is a deoxyribose if it's DNA, and an N base, okay? Hydrogen bonds hold them together, one nucleotide to the other. So A would represent the phosphate. B, because you see a U here, the B would be ribose. B is sugar. If it's DNA, it's deoxyribose. If it's RNA, it's ribose. C would be an N base. D is your hydrogen bonds, which holds one side of a nucleotide to the other, or connects the N bases. So DNA synthesizes in the nucleus of a cell during the S phase of the cell cycle. Which of the following does not describe RNA? Does RNA have an N base? Yes. Is RNA double helix? No. Does RNA have ribose sugar? Yes. RNA can leave the nucleus, and RNA has U instead of T in their nitrogenous bases. 5-carbon sugar is always attached to a phosphate, but it's also attached to an N base, so the best answer would be B. Okay? Because you have a 5-carbon sugar is like your little house, which is attached to a phosphate and attached to an N base. So it's always attached to both. When DNA makes a copy of itself, this process is called replication. Synthesis is also a word. This is not, okay? So it's all except B. RNA is made also in the nucleus of the cell. I'll type that for your reference. And then it can leave the nucleus, but it's made in the nucleus of the cell. The proteins, the actual protein is made in the cytoplasm, but the protein production starts in the nucleus. So this is not really the best question. What part of the cell is used to make protein is a better question, and the answer is both. What is responsible for protein production? Well, the start is DNA. You got a DNA strand, and it splits, and it makes RNA. And then the RNA goes and finds a ribosome to assist it in the making of the protein. So the answer is all of the above. In which part of the cell does transcription take place? Transcription is the first part of protein synthesis, so it happens in the nucleus of the cell. In case I don't say this later, the second part of protein synthesis is translation, and this happens in the cytoplasm. All right, so what are the corresponding bases? It would be T. I can't see, so I'm going to bring this down. G, C, A, T, C, G, T. And then I'll bring this up and line it up, and it should add up perfectly. So A with T, C with G, G with C, T with A, and there you got your corresponding end bases. For RNA, you can literally come back here, copy this. I'm going to come back over here, paste, and all I'm going to do is change my T's for U's because RNA does not have T's. And there's the answer for the next question. This is an example of a drawing of protein synthesis. So if we were going to label all of these parts, A would be DNA, B would be RNA, C would be tRNA, D would be anticodon, E would be ribosome, F would be codon, G would be mRNA, it's not really supposed to be all caps, but we'll just leave it at that. T would be amino acids. H, I'm sorry. I would be cytoplasm. And J would be the nucleus of the cell. Now, everything that happens in the nucleus is transcription, and everything that happens outside the nucleus is translation. To go through this process, DNA unzips and creates RNA. RNA gets modified and leaves the nucleus, becoming messenger RNA. Messenger RNA holds information in what we call codons, which is every 3 nucleotides. tRNA comes in based on this message, bringing a specific amino acid, and it comes in with the corresponding nitrogen bases, which are called anticodons. And each one of these tRNA brings in an amino acid, which eventually will form the protein chain as you see forming here. RNA is made during transcription. tRNA brings in the amino acids. RNA is made from DNA. The codons are found on mRNA. mRNA leaves the nucleus. tRNA holds the anticodon. Amino acids combine to form protein. Amino acids are the building blocks of protein. The backbone of DNA can be a phosphate and a 5-carbon sugar, which is also called deoxyribose. So it would be all except B. This would be the correct answer. Adenine always combines with thymine, and cytosine always combines with guanine, but adenine will temporarily combine with uracil when forming RNA. DNA makes protein, yes, in conjunction with RNA and ribosomes. Makes more DNA, yes, that process is called replication. DNA makes RNA, yes, in the first step of protein synthesis, called transcription. DNA makes amino acids, that is no. And the answer is all except D. DNA calls, or when DNA makes RNA, RNA calls for a specific amino acid, and then it builds the protein. Corresponding end bases again, we'll come over here, and the answer is A, T, A, T, C, C, G. And that's if you're making DNA. I think this question said put these in order. So we're going to go with unzips. mRNA is made. mRNA leaves the nucleus. mRNA attaches to a ribosome. The code is read every 3 letters, and they're called a codon. and tRNA brings the anticodon. Sorry, that would be a 6. And that's it. Guys, make sure to study also mutations. There's only like 3 terms you have to know there. Frame shift and just basic concept of mutations. Also, remember that enzymes are responsible for, and I'm going to come over here, or maybe over here where there's a picture. Enzymes are responsible for unzipping DNA. DNA is held together by hydrogen bonds, so the N bases are held together by hydrogen bonds, and enzymes come in and break these bonds, splitting the DNA or causing it to uncoil, both in replication and protein synthesis. Also, remember that there's a specific amino acid for a specific codon, so be familiar with that chart I gave you of the 64 codes, and they're specific. I'm going to include the chart on the test, but be familiar on how to read it. If you have any questions, just send me an email.